Last time we reviewed how RNA viruses make messenger RNA and RNA genomes. In this lecture, we're going to consider how DNA viruses make messenger RNA, a process that is called transcription. During replication of viruses with DNA genomes, viral messenger RNA synthesis must precede production of proteins. It is, in fact, the first biosynthetic event that takes place in cells infected with many DNA viruses genomes that have to be repaired, as we'll see in a few moments. So in this lecture, we're going to talk about viruses that make mRNA from DNA templates, and these will include double-stranded DNA viruses like adenoviruses, herpesviruses, and polyomaviruses. We'll also consider the parvoviruses and the hepatitis B viruses, which have genomes that are not double-stranded, and we'll talk about what happens to those. And we're also going to briefly consider retroviruses because their plus-stranded RNA genomes are reverse transcribed to a double-stranded DNA copy. There are three DNA-dependent RNA polymerases in cells, and they're called polymerases 1, 2, and 3, or Pol 1, 2, and 3, and they synthesize different kinds of cellular RNA. RNA polymerase 2 makes precursors to mRNA, as well as precursors of small regulatory molecules. RNA polymerases 1 and 3 make stable RNAs like ribosomal RNAs and transfer RNAs, respectively. For most DNA viruses, transcription is accomplished by Pol 2. This enzyme also transcribes proviral DNA of retroviruses. That means the retroviral DNA that's integrated into the cellular DNA. The signals that control expression of the genes of these viruses are therefore similar to those that control cellular gene expression. Now, in contrast to all these viruses, the genomes of pox viruses are transcribed by a viral DNA-dependent RNA polymerase, as well as other viral proteins that control recognition of viral promoters. We're not going to talk much about the pox viruses in this lecture or much in this course, but they do things very differently. Expression of viral genes is in a strictly defined reproducible sequence is a characteristic of cells infected by DNA viruses. In general, enzymes and regulatory proteins are made during the initial period of infection, and structural proteins are made only after viral DNA synthesis begins. This orderly pattern is primarily the result of transcriptional regulation by viral proteins. And this differs from the continual expression of all viral genes that's characteristic of the infectious cycles of many RNA viruses. Now, here on this slide, you can see the origin of the transcriptional component for different viruses. The simplest retroviruses use all host transcriptional components, not only Pol 2, but also ancillary proteins, as we'll see. And then a number of viruses use host proteins plus one viral protein that regulates transcription. And these include complex retroviruses, parvoviruses, papilloma, and polyomaviruses. And then the more complex DNA viruses, adenoviruses, and herpesviruses use host proteins plus more than one viral protein that regulate transcription. And as I just said, the pox viruses, the transcriptional components are entirely viral. All viral DNA molecules that are transcribed by cellular RNA polymerases must enter the cell nucleus. Some of these are double-stranded DNA molecules that can be transcribed as soon as they reach the nucleus. And so for those, adenoviruses, herpesviruses, papilloma, and polyomaviruses, transcription of specific genes is the first biosynthetic reaction in cells. Viral DNA synthesis is not required prior to transcription, but this process dramatically increases the concentration of templates and is actually crucial for the transcription of specific viral genes. Other viral genomes have to be converted to double-stranded molecules before they can serve as transcriptional templates. An example is the Adenovirus genome, which we've mentioned, which has a gap, a protein on one end, piece of RNA another. This has to be repaired so that it's fully double-stranded before it can be transcribed. The parvovirus genome is single-stranded DNA. It has to be repaired to be double-stranded before it can be transcribed. The retrovirus genome, which is a plus-stranded RNA genome, is converted to double-stranded DNA, which then integrates into the cellular DNA and serves as a transcription template. No matter how and where viral RNAs are made, they all have to be translated by the protein synthesizing machinery of the cell. And a series of covalent modifications that are called RNA-processing give mRNAs the features that are needed to be recognized by the protein synthesis machinery and to be translated by ribosomes. Two important modifications are the addition of a cap to the 5' end and the addition of multiple A residues to the 3' end. Those are called capping and polyadenylation. The enzymes that carry out these modifications can be viral or cellular. In addition, when an RNA is produced in the nucleus, another chemical rearrangement called splicing is possible. When viral RNAs are produced in the nucleus, it has to be exported to the cytoplasm for translation. This export is considered to be part of mRNA processing, even though the RNA is not known to undergo any chemical change during its transport. Viral mRNAs leave the nucleus by cellular pathways, but the cargo that is transported can be altered in virus-infected cells. So all of these processing reactions, which are shown here, not only produce functional RNAs, messenger RNAs, but they also provide numerous opportunities for post-transcriptional control of gene expression. The regulation of processing can increase, for example, the coding capacity of the viral genome. It can determine when specific proteins are made during infection, and also can help selectively express viral genetic information. The transcription of cellular and viral genes requires not only the template-directed synthesis of RNA, but also correct interpretation of DNA signals that mark the start and the stop of transcription. The initiation of transcription involves recognition of the point at which copying of the DNA should begin, and that's called the initiation site, which is shown in red here, as well as synthesis of the first few phosphodiester bonds in the RNA. Purified RNA polymerases can perform elongation reactions in vitro, but they are not capable of specific initiation of transcription without the assistance of additional polymerase-specific proteins. And both local and distant sequences can control transcription from core promoters, although in many cases local sequences are sufficient for proper regulation. These local regulatory sequences, which are shown on this slide, are recognized by sequence-specific DNA-binding proteins. Accurate initiation of transcription by Pol2 is directed by DNA sequences located near the site of initiation called the promoter. The promoter and the set of DNA sequences that control transcription make up the transcriptional control region. For Pol2, the initiation of transcription is a multi-step process, and some of these reactions include recognizing the promoter, that specific promoter sequence, unwinding of the DNA around the initiation site so that the polymerase can have access to the strand, and movement of the transcribing complex away from the promoter. At least 40 proteins, including Pol2 itself and initiation proteins, are needed to complete the process of initiation. The core promoters, which are shown here, contain all the information that's needed to recognize the site of initiation and to assemble a pre-initiation complex. These assemblies consist of RNA polymerase II and a set of general proteins, or called initiation proteins, that are needed to begin transcription. A hallmark of many core RNA polymerase promoters is the presence of a TA-rich TATA sequence, about 20 to 35 base pairs upstream of the site of initiation, and that is recognized by the TATA binding protein, TBP or TF2D. Short sequences termed initiators, which specify accurate but inefficient initiation of transcription in the absence of any other promoter sequences, are also commonly found in promoter regions. Transcription of viral genes is also regulated during the single-cell life cycles of many DNA viruses. Large quantities of viral proteins for assembly must be made within a very short infectious cycle, so some viral genes have to be transcribed at higher rates than others. In many cases, viral genes are transcribed in a specific temporal sequence, and this regulated transcription is achieved by the transcriptional control region, shown here for 3 different viruses, that contains binding sites for cell proteins. You can see, for example, the Adenovirus major late promoter has initiation site and also sequences within the promoter region for the binding of various proteins, as SV40 does. It's slightly different, and the Adenovirus early promoter is even different. So these utilize cellular promoters as well as viral proteins. The viral proteins that are made contribute to the regulation so that they can establish orderly transcription of viral genes. Efficient transcription of many viral and cellular genes also requires more distant regulatory sequences in the DNA template. And the first example of this was discovered in the genome of SV40. It was termed an enhancer. It's shown here in orange on this DNA because it stimulated transcription. Enhancers are defined by their position and orientation independent stimulation of transcription over distances as great as 10,000 base pairs in the genome. They can be 10,000 base pairs away from the initiation site. Enhancers are built with binding sites for proteins that recognize promoter sequences. And one of the models for how they work is called the DNA looping model, and it predicts interactions among enhancer-bound proteins and the transcriptional components assembled at the promoter with the intervening DNA looped out. So here we see the assembly of Pol2, which is shown in pink, and all the other proteins that are required to start initiation of transcription. And this enhancer region is tethered to this by these enhancer binding proteins, and this makes transcription more efficient. All viral templates that are transcribed by Pol2 of the cell contain transcriptional control regions that are directly recognized by cellular regulatory proteins, and we've seen some images already of the binding sites for these in the promoter regions. These cellular sequence-specific transcriptional regulators play important roles in the expression of viral genes. However, the genomes of many viruses also encode transcriptional regulatory proteins, and some of those resemble cellular proteins that recognize local promoter or more distant enhancer sequences. The cellular and viral DNA binding proteins that are needed for transcription from viral DNA templates share a number of common properties. Their most characteristic feature is modular organization, such as shown here. They are built from discrete structural and functional domains. And the basic modules are a DNA binding domain, which does exactly as its name suggests, and an activation domain, which helps to make transcription more efficient. And these can function independently. They can be swapped among different proteins. You can also have a domain that specifies dimer formation, because often these proteins bind to DNA as dimers. The regulation of transcription by these sequence-specific DNA binding proteins usually requires additional proteins that are called co-activators or co-repressors. These proteins typically can't bind specifically to DNA, and they can't modulate transcription on their own. But once they're recruited to a promoter, for example, by binding to a sequence-specific regulator that's already bound to DNA, they can augment or dampen transcriptional responses. A common feature of many such co-regulators is their ability to alter the structure of nucleosomal templates for transcription, either directly or by interaction with appropriate enzymes. And I think you know that chromosomal DNA is ordered in nucleosomes, and the structure of those nucleosomes, whether they're tight or loose, can control transcriptional activity. So let's take a pause here and answer a question. Which of the following is correct concerning proteins that regulate transcription? 1, they include sequence-specific activator proteins that bind promoter elements. 2, they include co-activators, which do not bind DNA. 3, some viral genomes encode co-activators. And 4, they may have multiple protein domains. And 5, maybe all of these are correct. And you might want to pause the recording at this point because I'm going to give you the answer. And the answer is number 5. All of the above are correct. They include activator proteins that bind promoters. They include co-activators, which don't bind DNA, just define that. And some viral genomes can encode co-activators, and they can have multiple protein domains, as we've just discussed. The transcription of many viral DNA templates by RNA-Pol2 results in the synthesis of a lot of transcripts, sometimes over 100,000 copies of an individual mRNA species in a cell in a pretty short period of time. These bursts of transcription are basically elicited by transcriptional regulatory proteins. And viral proteins that stimulate Pol2 transcription establish one of two kinds of regulatory circuit. The first is a positive autoregulatory loop, in which a viral activating protein stimulates the rate of transcription but doesn't alter the nature of viral proteins that are made in cells. Now, just a corollary, there are also negative autoregulatory loops which repress transcription, but here we're talking about augmenting transcription. And the second is a transcriptional cascade in which different viral transcription units are activated in a fixed sequence. This mechanism ensures that different classes of viral proteins are made during different periods of the infectious cycle. And as I've already mentioned, this is characteristic of viruses with DNA genomes. So you can see that the participation of viral regulatory proteins gives a measure of control that's not possible when the transcriptional program is carried out only by cellular components. Now here's just an illustration of these two types of regulation. At the top is a positive autoregulatory loop. Here we have a DNA that encodes a protein. It's shown by gene X. The promoter for that protein, for that gene, results in the production of an mRNA and eventually a protein. And that protein happens to be a transcriptional activator, which can stimulate in a positive way the transcription of the same gene. So it's a positive autoregulatory loop. You can make more mRNA of the same protein. And you can imagine a negative loop would be that the gene product would be repressing transcription. On the bottom is an example of cascade regulation. Here we have a double-stranded DNA genome encoding two genes X and Y. In this case, the production of X and its protein product is necessary for the transcription of gene Y. Without protein X, gene Y is not made. So that's cascade regulation. A striking characteristic of the productive infectious cycles of DNA viruses is transcription of viral genes in a reproducible and precise temporal sequence. And before initiating genome replication during what we call the immediate early and early phases of infection, cells infected by viruses synthesize viral proteins that are needed for viral DNA synthesis, efficient viral gene expression, or maybe other regulatory functions. The transcription of late genes, most of which usually encode structural proteins, requires genome replication. This property ensures coordinated production of the DNA genomes and the structural proteins from which progeny virus particles are assembled. Another common feature is the control of the transitions from one transcriptional stage to the next by both viral proteins and genome replication. These viral programs closely resemble those that regulate many developmental processes in animals, in both the transcription of individual genes in a predetermined sequence and the sequential action of proteins that regulate transcription of different sets of genes. Now, as shown on this side, we have three different viral genomes and the transcriptional programs that are representative of each. And you can see that the sophistication of the transcriptional regulatory circuits increases with genome size. Here at the top we have SV40 genome, where the synthesis of a single viral protein, large T, in infected cells leads inevitably to entry into the late phase of infection. And that's because T antigen both induces initiation of viral DNA synthesis and activates late transcription. So large T is made early in infection. Eventually it represses its own synthesis, but it in turn activates viral DNA synthesis. It activates late transcription, and DNA synthesis also is an activator of late transcription as well. Now, in contrast to this simple system with one protein, at least 3 viral transcriptional regulators control the transitions from one phase to the next during the adenovirus infectious cycle. The immediate early, i.e. E1A proteins, are needed for efficient transcription of all early transcription units. Among this set is the E2 gene, which encodes proteins needed for DNA synthesis, relief of repression of transcription of the gene encoding the sequence-specific DNA-binding protein 4A2, and activation of transcription from the major late promoter. And this promoter controls the synthesis of most of the viral structural proteins. The synthesis of progeny viral DNA is therefore coordinated with the production of the proteins that will eventually encapsulate the genome. There are even more complicated coordinated transcriptional programs exemplified by herpes simplex virus. And here, multiple transcriptional programs are required to get into the late phase, and we'll consider this in a subsequent slide. Now let's look at the transcription cycle of Polyomavirus. This is a rather simple genome made up of double-stranded DNA. It has a single transcription initiation site from which all the mRNAs are made. And we have an early phase, which is synthesis of mRNAs shown to the left here. They initiate from the red box, which is labeled ORI. And you can see you have multiple mRNAs, and splicing gives you several different protein products, including the T antigen. And then there's a late phase, which encodes, again, a number of mRNAs, and these are mainly for structural proteins. So we have 2 phases. They're regulated by large T antigen. There are not many regulatory sequences, just around the origin of replication. And the mRNAs, the different mRNAs, are made by differential splicing and polyadenylation. Now back to adenoviruses. They encode multiple transcriptional regulators. The immediate early E1A protein is needed for transcription of all the early transcription. E1A is needed for early transcription units, and then one of the products of the early transcription units, E2, is needed for DNA synthesis and the entry into the late phase, and it also increases initiation from the major late promoter, so that then we can have transcription of the 4A2 gene, which will also enhance the late gene transcription. Now, on the top, I've shown you the adenovirus genome as a single line, but it's obviously a double-stranded DNA, and I just want to show you that to give you an idea of the complexity of these transcriptional units. Both strands of DNA serve as templates for mRNA synthesis, so you can see some mRNAs are transcribed from the top, and some are transcribed from the bottom. And the green mRNAs represent Pol2 products, and the tan ones represent Pol3 products. You can see their early transcription units. They start with an E. There are a few up here on the left. Here's an E3 and E4. And then there are later transcription units, such as the major late promoter, which drives the synthesis of mRNAs encoding structural proteins. And so these are all coordinately expressed, as we've shown before, just so that you have a precise timing of the products when you need them. So here the whole system of adenovirus replication is depicted in the replication cycle. We've talked about how these viruses get into cells. Eventually the genome is imported in the nucleus. Pol2 then transcribes the E1A gene, the immediate early E1A gene, and these proteins are made, of course, in the cytoplasm. They're then imported back into the nucleus, where they regulate the transcription of the early genes. And the larger version, you can see there are 2 different sizes here. The larger version of E1A stimulates transcription of viral early genes by cellular Pol2. There's also transcription of one of the viral genes by Pol3 early in infection. Early proteins include viral replication proteins, which are imported into the nucleus, like the polymerase DNA binding protein and so forth, which we'll talk about later. and these cooperate with some cellular proteins to replicate the viral genome. And the replicated viral DNA molecules can be templates for more DNA or transcription of the late genes. Some late promoters are activated simply by viral DNA replication, but maximally efficient transcription of the major late transcription unit requires 4A2 proteins and the L4 protein. There are over 80 known protein coding sequences in the herpes simplex virus genome, and these are, with few exceptions, expressed as individual transcription units. The basic distinction of early and late phases is maintained in the herpesvirus transcriptional program, but temporal control of the activity of more than 80 viral promoters is more complicated. But obviously you can finely tune regulation to a much greater extent when you have so many independent transcription units. For adenoviruses and polyomaviruses, cellular enhancer and promoter binding proteins are enough to initiate the transcription programs. So the T antigen of SV40, the immediate early protein of adenovirus, they can all be produced from mRNAs transcribed using the cellular transcriptional machinery. In contrast, a viral activating protein imported into the cells within the virus particle, it's called VP16, is essential for efficient transcription of herpes viral immediate early genes. So unless this protein is brought in with the virion, no gene expression will occur because the cell transcriptional proteins, the cell transcriptional machinery, cannot recognize this immediate early promoter. The regulatory scheme of herpes simplex virus is even more complex than the two we've talked about. The immediate early gene products include two transcriptional regulators, ICP4 and ICP0. The ICP4 protein, like the E1A proteins of adenovirus, is essential for progression beyond the immediate early phase of infection. It's considered to be the major essential transcriptional activator. It stimulates the transcription of both early and late genes and also acts as a repressor of immediate early transcription. Some herpes virus late genes are transcribed only following synthesis of progeny genomes, the pattern that was exemplified by SV40 late transcription, and other promoters attain their maximal rates of transcription during the late phase. So again, we have the similar cascade of reactions. Immediate early transcription is initiated by VP16, a virion-associated protein. It activates immediate early transcription, and 4 and 0 are the products of that. The immediate early proteins control transcriptions from all virus genes, the early genes, DNA synthesis, and late genes, and this ensures a coordinated production of DNA genomes and structural proteins. And to put this in the context of a viral replication cycle, the viral particle is entering the cell here, and the key point is that VP16 is coming out of the particle as the viral membrane fuses with the cell and diffuses into the nucleus. And then when viral genomes enter the nucleus, these VP16 molecules can activate transcription of immediate early genes, and that's shown here. So those mRNAs go out into the cytoplasm. The immediate early proteins, also called alpha proteins, are made. They come back in the nucleus. They stimulate the production of early proteins or beta proteins, which can then come into the nucleus and stimulate the replication of DNA. And the DNA replication, among others, is needed for the efficient expression of late mRNAs and the late proteins, which are structural proteins needed to build new virus particles. Let's take a moment for another quiz. This question is adenovirus E1A protein stimulating the expression of adenovirus E2 protein, which then stimulates the expression of adenovirus 4A2 and L4 protein, is an example of 1, a negative autoregulatory loop, 2, repression of gene expression, 3, cascade regulation, 4, dimerization. And you should stop the recording at this point because I'll tell you the answer, which is, of course, 3. It's cascade regulation, transcriptional events happening in a precise, regular sequence. Now we're going to talk about some of the modifications that occur to these messenger RNAs as they are being produced in the nucleus that are essential for their function. And one of them is the cap structure and the other is polyadenylation. Virtually all viral and cellular mRNAs have the same cap structure, M7GPPN. So what that means is N is any nucleotide, the first base here. The cap is usually a G. It can be methylated on the seventh residue of the ring. And it is connected in a 5' to 5' linkage to the first base rather than in a 5' to 3' linkage, which is typical. This kind of structure protects mRNAs from 5' exonucleases degrading it, and it's also essential for efficient translation of most mRNAs because it's recognized by the translation initiation machinery. The cap also blocks recognition of viral RNAs by an antiviral defense mechanism that detects cytoplasmic RNA molecules carrying uncapped 5' triphosphate groups. Now, although most viral RNAs have a 5' cap structure, there is variation in how the modification is put onto the RNAs. And we can see three mechanisms: de novo synthesis by cellular enzymes, synthesis by viral enzymes, and acquisition of preformed 5' cap structures from cellular mRNAs, which is what mechanism we talked about last time when we discussed cap snatching by the influenza RNA polymerase. Viral pre-mRNA substrates for the capping enzyme are made in the infected cell nucleus by Pol2, and the formation of caps on the 5' ends of both viral and cellular transcripts, which is the first step in their processing, is a co-transcriptional reaction that takes place when the nascent RNA is only about 20 to 30 nucleotides long. So on this DNA with a initiation sequence, we're showing an RNA polymerase beginning to make an RNA transcript. It's 20 to 30 nucleotides long. Then the RNA polymerase is phosphorylated at specific serines in the C-terminal domain of the largest subunit, and that's the signal for binding of the capping enzyme, and which then caps the nascent RNA. The close relationship between the capping enzyme and Pol2 makes sure that all the transcripts are capped. And that also explains why RNA species made by the other polymerases 1 and 3 are not modified. They do not carry an equivalent C-terminal domain as Pol2 does. Just like the 5' cap structure, the 3' poly-A sequence on mRNA was first identified in a viral mRNA. And that modification was soon found to be a common feature of mRNAs made in cells that were not infected. Like the 5' cap, the 3' poly-A sequence stabilizes mRNA, and it also increases the efficiency of translation. The addition of 3' poly-A to viral pre-mRNAs, just like capping at the 5' end, can be done either by cellular or viral enzymes. Now, viral pre-mRNAs made in infected cells by Pol2 are polyadenylated by cellular enzymes. Transcription of a viral or cellular gene by RNA Pol2 proceeds beyond the site at which Pol2 will be added, which is right here. And you can see the transcript goes beyond it. The 3' end of the mRNA is determined by endonucleolytic cleavage of its pre-mRNA at a specific position. And that cleavage is also required for terminating transcription. Poly-A is then added to the new 3' terminus, while the RNA downstream of the cleavage site is degraded. Cleavage and polyadenylation sites are identified by specific sequences. The 3' end of mature mRNA is formed 10 to 30 nucleotides downstream of the highly conserved and essential polyadenylation signal AAUAAA. This sequence is not enough to specify poly-A addition, however. Sequences 3' of the cleavage site, such as an A or GU-rich sequence located 5 to 20 nucleotides downstream, are also needed. In many mRNAs, especially viral mRNAs, additional sequence 5' of the cleavage site is also important. So the first reaction in polyadenylation is recognition of the AAUAAA by the protein called CPSF, which stands for cleavage and polyadenylation specificity protein, and that interaction is stabilized by other proteins. Poly-A polymerase is then recruited to the complex, and following cleavage of the pre-mRNA, that enzyme synthesizes a poly-A segment of 200 to 250 nucleotides long in a two-step process, a slow and then a rapid polyadenylation. Components of the polyadenylation machinery like capping enzymes are also associated with Pol2, and the polymerase is required for efficient polyadenylation. Both CPSF and CSF are essential, bind to the C-terminal domain of the large subunit of RNA Pol2. And these interactions are required for polyadenylation and are believed to coordinate the synthesis of pre-mRNAs with maturation of the 3' ends. So this is a summary of the mechanisms by which poly-A is added to viral RNAs. We've just discussed post-transcriptional mechanism, which occurs when DNA virus mRNAs are transcribed by cellular enzymes, cleavage of pre-mRNA followed by polyadenylation. The enzyme that does this is cellular, and these are the viruses that do this. And this is in contrast to polyadenylation in RNA viruses that we talked about last time. For example, influenza virus and VSV, reiterative copying at stretches of U in the template RNA. And this, of course, is carried out by the viral polymerase. And then for poliovirus and togoviruses, copying of a long stretch of U in the template. So, for example, there's a stretch of U at the 5' end of the minus strand, which becomes an A stretch or poly-A in the plus strand. That's also done by a viral polymerase. So let's do another question. Which statement about polyadenylation of DNA virus mRNAs is correct? 1, it always occurs in the cytoplasm. 2, it occurs after cleavage of pre-mRNA. 3, poly-A is added at the 5' end of pre-mRNA. And 4 is specified by a stretch of U residues in the template. So why don't you pause the recording while I give you the answer. And, of course, it occurs after cleavage of pre-mRNA. It doesn't occur in the cytoplasm. This is a nuclear event. Poly-A is added at the 3' end where the cleavage site is, and it is not specified by a stretch of U. That's true for some RNA viruses, but the question asked, of course, about DNA viruses. Between 1960 and the mid-1970s, scientists who were studying nuclear precursors of mammalian mRNAs found that these RNAs are larger than the mRNAs that were translated in the cytoplasm, and they're also heterogeneous in size. So they were therefore named heterogeneous nuclear RNAs, or hnRNAs. These hnRNAs were shown to carry both the 5' cap structure and 3' poly-A sequence, which led to the conclusion that both ends were preserved. Both ends of the hnRNA were preserved in the small, mature mRNA. So basically, investigators had this conundrum of figuring out how small RNAs could be produced from larger ones when both ends were maintained. And this puzzle was solved with the discovery of pre-mRNA splicing. And this was done by two groups of investigators, one led by Philip Sharp, another by Richard Roberts, and they both shared the 1993 Nobel Prize in Physiology or Medicine for this work, which showed that the major late mRNAs of adenovirus are encoded by 4 separate genomic sequences. The distribution of the mRNA coding sequences into 4 separate blocks in the genome, in conjunction with the large size of the major late mRNA precursors, implied that these RNAs were synthesized by excision of non-coding sequences from primary transcripts, called introns, with precise joining of coding sequences, or exons. The demonstration that nuclear major late transcripts contain the non-coding sequences, or introns, confirmed that mature RNAs mRNAs are formed by splicing of non-contiguous coding sequences in the pre-mRNA. This mechanism for mRNA synthesis had great appeal because it could account for the puzzling properties of hnRNA. Within a matter of months, it was shown that splicing of pre-mRNA is not an obscure virus happening, but it happens in all eukaryotic cells. The great majority of mammalian pre-mRNAs, like adenoviral major late mRNAs, have exons interspersed among introns. So in this slide, it summarizes the data that led to the conclusion that mRNAs in adenovirus infected cells are spliced. On the left, we have the major late mRNAs, which is a mixture of different mRNAs of different lengths, all made from the same promoter. These are digested with RNase T1, which cuts after a G residue, and then the capped oligonucleotides were isolated. And what they found was that the same 11 nucleotide sequence was present at the 5' ends of all the late mRNAs, and that was a bit puzzling to the investigators. Hybridization studies showed that these 5' ends were not encoded next to the main segments of the major late mRNAs. So what was then done was to directly visualize mRNAs hybridized to viral DNA, and that provided proof that the coding sequences are dispersed. So in other words, this 11 nucleotide sequence isn't simply present in front of all the bodies of the different mRNAs. So here's the diagram of this experiment. It's one major late mRNA encoding the hexon protein, and that's shown in green here, and it's hybridized to a complementary adenovirus DNA fragment extending from the left end of a genome to a point within the hexon coding sequence. So you can see the DNA is looping out. It's not hybridizing all along the length of the hexon mRNA. So we have 3 loops, which we see, and they're designated A, B, and C, bounded or separated by 3 short segments, 1, 2, and 3, and 1 long segment, hexon RNA of DNA-RNA hybrid. The DNA-RNA hybrid is the thick lines. So this same pattern was observed for other adenovirus late mRNAs, but they differed in the length of this loop and the length and location of the DNA-RNA hybrid. And they concluded from this data that the major late mRNAs contain a common 5' terminal segment, segments 1, 2, and 3, which are built from sequences encoded at 3 different sites in the genome. And they call this the tripartite leader. This sequence is then joined to the mRNA body, and a long sequence, which is a long sequence complementary to the hexon coding sequence in the viral genome. So here at the bottom is a schematic of the gene encoding this mRNA. So here's the adenovirus genome. The tripartite leader sequence is in 3 pieces, and the hexon mRNA body is shown in green. And the blue and the orange and the red parts are spliced out so that you join up these 3 black ones together and join it to the hexon body. These 3 tripartite leader sequences are only present at the very end here of the gene, and they're spliced in turn to the different major late mRNAs to give them each a 5' end composed of this sequence. Sequencing of DNA of a large number of cellular and viral mRNAs and their genes identified short consensus sequences at the 5' and 3' splice sites, which are joined to each other in mature mRNA. So here is a diagram of a pre-mRNA. The mature RNA is in green, and the pink is what is spliced out. So here's the 5' splice site, which will be joined to the 3' splice sites. And below this are the frequency of occurrence of these bases. The conserved sequences lie largely within the introns, as you can see. And the dinucleotides GU and AG are found at the 5' and 3' ends. They're 100% conserved on all introns. Mutation of any of those 4 nucleotides eliminates splicing, so they're essential for splicing. Then there's one in the middle here that's also 100% conserved. So the way pre-mRNA splicing occurs is by two trans-esterification reactions in which one phosphodiester bond is exchanged for another one without the need for an external supply of energy. The first reaction yields two products, the 5' exon and the intron 3' exon lariat. So you have an attack of the hydroxyl of this A residue on the phosphorus here, and that's why that A residue is conserved, because that's carrying out the attack. So you now have this looped sequence form. It's attached to the 3' exon, and then the 5' exon is free. And the second reaction, the newly formed hydroxyl group at the end of the 5' exon attacks the phosphodiester bond at the 3' splice site. You now have the spliced product and the excised lariat intron. Chemically, this splicing is a simple process, but think about it. Each splicing reaction has to be completed with a high degree of accuracy to make sure coding sequence is not lost, and the chemically active hydroxyl groups have to be brought into close proximity to the phosphodiester bonds that they're going to attack. And many genes have very large introns separating multiple exons, and they have to be spliced in the correct order. For this reason, pre-mRNA splicing occurs in a very large structure called the spliceosome, which contains many proteins and small RNAs, and these associate with the pre-mRNA and ensure that these reactions all occur properly. 5 different small nuclear RNAs, or snRNAs, participate in splicing. They're called U1, U2, U4, U5, and U6, and you can see them on this figure. In vertebrates, these these RNAs vary in length from 100 to 200 nucleotides, and they are associated with proteins to form what are called small nuclear ribonuclear proteins, or SNRPs. And you can see there are these shaded areas of the proteins here. The RNA components of the SNRPs recognize splice sites and other sequences in cellular and viral pre-mRNAs. They participate in multiple interactions with the pre-mRNA and also with each other during both the assembly of the spliceosome and splicing. Assembly of the spliceosome results in base pairing of the snRNA components of the U1 and U2 snRNPs with the 5' splice site and the intronic branch point, respectively. Rearrangements of the RNA-RNA interactions within the spliceosome then result in juxtaposition of the 5' splice site and the branch point for the first transesterification reaction. The RNA of the U5 snRNP juxtaposes the 5' and 3' ends to facilitate catalysis of their joining in the second transesterification reaction. Now, these RNAs of the SNRPs do more than organize the pre-mRNA sequences as shown by the interactions on this slide. The spliceosome is in fact an RNA enzyme or a ribozyme, and there's compelling evidence that the U6 SNRPs has catalytic activity during splicing. Although the RNAs of SNRPs play essential roles in splicing as guides and catalysts, the spliceosome also has non-snRNP proteins. One of them comprises proteins that package the pre-mRNA substrate. These are called heterogeneous nuclear ribonuclear protein, HNRNP proteins. And many other splicing proteins contain both RNA binding and protein-protein interaction domains. These proteins bind to pre-mRNA sequences within or adjacent to exons to help spliceosome assembly or to regulate splicing and exon recognition. Other proteins that are important for splicing are helicases, which are thought to catalyze the rearrangements of hydrogen bonding among the different snRNAs in the pre-mRNA substrate. And all of this is co-transcriptional. Splicing is co-transcriptional, and components of the splicing machinery associate with that hyperphosphorylated form of the C-terminal domain of the large subunit of Pol II during elongation. Now you may be wondering, why are genes spliced? Why are mRNAs spliced in this way? There are lots of reasons for them, and here are just three. Alternative splicing, which we'll define in a moment, creates new functional genes. The coding information of a small DNA genome can be expanded by splicing, and it can also be used to regulate gene expression. Many viral and cellular pre-mRNAs contain multiple exons, as shown here. So the exons are in green, the introns are in pink. The introns are going to go away, the exons are left to form the mRNA. Splicing of the majority removes all introns and joins all the exons in the order in which they're present in the pre-mRNA. This is called constitutive splicing, and it makes a single mature mRNA from this intron-containing mRNA. Many cellular and many viral pre-mRNAs yield more than one mRNA as a result of splicing different combination of exons in a process called alternative splicing. Several different types of alternative splicing occur. As this process generally leads to the synthesis of mRNAs that differ in their protein coding sequences, its most obvious advantage is that it can expand the coding capacity of viral genomes. For example, the early genes of polyomaviruses and the adenovirus E1A and E4 genes each specify 2 or more proteins as a result of splicing of primary transcripts at alternative 5' or 3' splice sites. And alternative splicing can also be important for temporal regulation of gene expression or controlling the balance in the production of spliced and unspliced mRNAs. And some examples of alternative splicing are shown here, where, for example, you can skip exons and get 2 different proteins. You use alternative 5' or 3' splice sites. So you expand the coding capacity and you allow for regulation of specific proteins at different times. Here's an example of how alternative splicing can be regulated. We're looking at the late proteins of bovine papillomavirus type 1. There's the double-stranded DNA genome. It's normally circular, but we're showing it as a linear DNA. These late proteins are synthesized efficiently only in highly differentiated keratinocytes. Productive replication of viral DNA and assembly and release of viruses are restricted to these outer cells in the epithelium. Alterations in splicing are crucial for production of the mRNA, encoding the major capsid protein called the L1 mRNA and the L2 mRNA. This mRNA is made only in fully differentiated cells and requires activation of alternative 3' splice sites that are not recognized in undifferentiated cells. The 3' splice sites for papillomavirus late mRNAs are suboptimal, and their utilization is governed by cis-acting suppressor or enhancer sequences in the pre-mRNA. It's been proposed that terminal differentiation of keratinocytes is accompanied by changes in the activity or abundance of cellular proteins that bind to these sequences. And in support of this idea, the hnRNP-A1 protein that binds to a sequence that inhibits recognition of the downstream 3' splice site of human papillomavirus type 16 late pre-mRNA is not present in terminally differentiated keratinocytes. So the DNA is shown here in linear form. The open reading frames are shown above. 2 of the RNAs made from the late promoter are shown to illustrate changes in recognition of splice sites and polyadenylation sites that are needed to produce the L1 mRNA. The synthesis of this mRNA depends on the recognition of a 3' splice site at position 3,605 rather than 3,225, which is used during the early phase of infection. Polyadenylation of pre-mRNAs must also switch from the early AE to the late polyadenylation site. On the right is in situ hybridization of bovine fibropapillomas using probes that specifically detect mRNAs spliced at the 3,225 site on the left or at the 3605 site on the right. Note the production of late mRNA spliced at the 3605 3' splice site occurs only in the outermost layer, as opposed to the early mRNA is produced in both layers. And below are the mechanisms that regulate splicing to produce this L1 mRNA, which are specific, again, to highly differentiated keratinocytes of the granular cell layer. The sequences that control alternative splicing are located between the two splice sites. The splicing enhancers SE1, SE2, and SE4 are recognized by cellular SR, serine, and arginine-rich, and other splicing proteins. The SE1 enhancer and the adjacent sequence that inhibits splicing at the 3605 5' splice site, termed exonic splicing suppressor 1, are thought to facilitate recruitment of U2-associated protein U2AF and recognition of the branch point sequence upstream of the 3225 3' splice site. SE2 is located very close to the 3605 3' splice site and may block access to the branch point for splicing at this site until keratinocytes differentiate. And here on panel D is shown inhibition of polyadenylation at the AE site by the binding of U1 snRNP to a pseudo 5' splice site located nearby in the primary transcript. And that inhibition is the result of binding of the U1 snRNP 70K subunit to poly-A polymerase. The expression of certain coding sequences in retroviral genomes, GAG and POL, for example, depends on an unusual form of alternative splicing that produces both spliced and unspliced mRNAs. Normally, all pre-mRNAs that contain introns are fully spliced in mammalian cell nuclei. In cells infected with retroviruses with simple genomes, like avian leukosis virus, a full-length, unspliced transcript of proviral DNA serves as both the genome and the mRNA for the capsid proteins and viral enzymes, while a singly spliced mRNA specifies the viral envelope protein. Retrovirus production depends on the maintenance of a proper balance between the proportions of spliced and unspliced RNAs. Modest changes in splicing efficiencies cause replication defects, as you can see in the table on the right. So normally the ratio of unspliced to spliced RNA is about 2:1 in wild-type virus-infected cells. And you can alter this ratio with mutations in the splicing signals. And these, you can see, reduce viral propagation. The features that maintain the proper splicing balance include a suboptimal 3' splice site and a splicing enhancer in the adjacent exon. A negative regulatory sequence located more than 4,000 nucleotides upstream of the 3' splice site is also important. This sequence, which is bound by both U1 snRNP and specific cellular protein, has been proposed to act as a decoy 5' splice site. It forms a complex with the 3' splice site for production of envelope mRNA, but one that does not participate in splicing reactions. We haven't talked about proviral DNAs much. We'll do that in a separate lecture, but the proviral DNAs of retroviruses contain long terminal repeat sequences at either end, and in each one is a poly-A addition signal. Transcription of some proviral DNA, like that of Rous sarcoma virus, initiates downstream of the long terminal repeat sequence, encoding the signals essential for polyadenylation. So you only get a poly-A signal at the 3' end. But many other retroviruses carry complete signals for this modification at both ends. So here you can see the initiation is happening upstream of this poly-A signal. Nevertheless, poly-A is added only to the 3' ends of these pre-mRNAs. There are two mechanisms to ensure that the correct poly-A addition signal of HIV type 1 pre-mRNA is recognized. Sequences present only at the 3' end of pre-mRNA stimulate polyadenylation by facilitating binding of CPSEP to the nearby AAU-AAA sequence. And in addition, recognition of the 5' poly-A signal is suppressed by a 5' splice site lying 195 nucleotides downstream. So here's the 5' poly-A signal. And you may remember a related mechanism, the binding of U1 SNRMP to a pseudo 5' splice site, also suppresses recognition of the poly-A addition site for the production of bovine papillomavirus type 1 late mRNAs until the keratinocyte host cell is fully differentiated, and that's when the virus particles are made. The production of adenoviral late mRNAs epitomizes complex alternative splicing and polyadenylation at multiple sites in a pre-mRNA, as shown here. The major late pre-mRNA shown here, covering most of the genome, contains the sequences of at least 15 different mRNAs. These fall into 5 families, L1 to L5, which are defined by which of 5 polyadenylation sites is recognized. The frequency with which each site is used must therefore be regulated to allow production of all late RNAs. For example, if you had high-efficiency polyadenylation at L1, you'd never get synthesis of the subsequent mRNAs. During the last phase of infection, each of these 5 polyadenylation sites directs 3' end formation with about the same frequency. All major late mRNAs also contain the 5' terminal tripartite leader sequence, which we've discussed before as being derived by splicing of the 3 sequences at the 5' end of the transcript. The splicing reactions that produce this sequence from the 3 small exons take place before polyadenylation of the primary transcript, and the final splicing reaction joins the tripartite leader sequence to one of many mRNA sequences. Each primary transcript therefore yields only a single mRNA, even though it contains the sequences for many, and most of the sequence is thrown away. It remains a mystery why this is such a bizarre mechanism for making adenovirus late mRNAs. And one idea is that it's a good way to ensure that every mRNA contains the 5' tripartite leader, which is important for efficient translation late in the infectious cycle. Any messenger RNA made in the nucleus has to be transported to the cytoplasm for translation. Other classes of RNA, like small cellular and viral RNAs made by Pol3, also have to enter the cytoplasm, transfer RNAs, small nuclear RNAs. This export is mediated by the host cell machinery, and in most cases it's indistinguishable from export of cellular RNAs. However, transport to the cytoplasm of unusual RNA molecules is a critical step in some viral life cycles. The substrates for mRNA export are not naked mRNA molecules, but rather ribonucleoproteins, shown here as RNA with proteins bound to it. All viral RNAs, mRNAs, made in the infected cell nuclei carry the same 5' and 3' terminal modifications as cellular mRNAs are exported. Many viral mRNAs, like their cellular counterparts, are produced by splicing. Cellular pre-mRNAs that contain introns and splice sites ordinarily are retained in the nucleus, at least in part, because they remain associated with spliceosomes, which is diagrammed here. In addition, a protein complex that marks mature mRNAs for export is assembled on the RNA only during splicing. As a result, pre-mRNAs are either spliced to completion and exported, or if they are not spliced, they're retained in the nucleus and degraded. so spliced RNAs are marked for export. However, the reproduction of retroviruses, herpesviruses, and some others requires production of mRNAs that are incompletely spliced or not spliced at all. So, how does that work? HIV-1 encodes a protein called REV. It's shown here in red. That protein promotes export of the unspliced and partially spliced viral RNAs. REV binds to an RNA sequence termed the REV responsive element that's within an alternatively spliced intron of viral pre-mRNA. This REV response element is 350 nucleotides long, and it makes several stem loops, one of which binds with high affinity the arginine-rich RNA binding domain of REV. The C-terminal domain of REV contains a nuclear export signal. One cellular protein that binds to the nuclear export signal of REV is exportin-1. Exportin-1 binds simultaneously to REV and a nuclear export protein called RAN. It is the receptor for REV-dependent export of the HIV RNAs that are bound to it. The viral protein therefore functions as an adapter, directing viral intron-containing mRNAs to a pre-existing cellular export receptor. So, for example, when the viral genome is expressed as an mRNA in cells, it's initially spliced completely to give a tiny mRNA that encodes the REV protein. The REV protein then goes back in the nucleus and binds to the REV responsive element in either unspliced or partially spliced mRNAs and enables them to be exported. If they weren't completely spliced, they would normally not be exported, but REV saves the day. However, many retroviral genomes do not encode REV proteins, yet their unspliced viral RNAs have to reach the cytoplasm. These unspliced viral mRNAs contain specific sequences that promote export called constitutive transport elements, or CTEs. So here is a CTE shown at the 3' end of an unspliced retroviral mRNA. The human nuclear export factor, NFX1, binds specifically to CTE and is essential for export of the RNA from the nucleus, and it is exported in an unspliced state. The direct and specific binding of NFX1 to the CTE of unspliced retroviral RNAs bypasses the cellular process, which ensures that export is normally strictly coupled with splicing. So here on the right, export of this mRNA is coupled with splicing because the spliceosome components mark it for export. The cellular export proteins like NFX1 recognize the splicing proteins and export only the spliced mRNA. The retroviral RNA that is not spliced has CTE, which will bind this export protein NFX1 and allows it to be exported. So unspliced or partially spliced RNAs can be exported from the infected cell nucleus by virtue of the CTE, which is recognized by the export machinery. And viruses always find a way to get around a limitation.